Okay, hi everyone. Today I'll be talking about DNA transcription, which is the synthesis of an mRNA strand. We only need to know one enzyme for this, which is the RNA polymerase, and we're looking at one length of DNA here, which that codes for a polypeptide, which is also known as a gene. So once the body receives signal to produce a protein, a bunch of transcription factors occur, which then enters the nucleus and starts off this process by first making the polymerase migrate to the promoter region of the gene. Once it's at the promoter region of the gene, it breaks the hydrogen bonds in between the bases, which unwinds the DNA. We now have two strands of DNA. One that's going to be used as the template, which is going to be this bottom one here, and that one is called the antisense strand, and the one on top is called the sense strand. So there's a lot of using the free ribonucleotides that's just kind of floating around. The RNA polymerase starts to synthesize the mRNA strand in the 5' to 3' direction. And this is done through complementary base pairing. So for example, if the antisense strand has the bases C, G, A, G, G, A, then the basis for the mRNA would be G, C, U, because RNA doesn't have thymine, it has uracil, and CCU. Then the polymerase just kind of continues along the gene and continues this process of unzipping and synthesizing mRNA. Again, always remembering from the 5' to 3' direction. And because of that, the direction of elongation will be in this direction, because the bases will go to this 3' end, but then the direction of unzipping happens in this direction. Eventually, the polymerase will reach an end that has a terminator region, and here it will dissociate or leave the DNA. It's no longer needed. And then we also have our synthesized mRNA strand. However, this strand is not yet ready to go to translation. mRNA is not ready for translation because DNA actually has coding and non-coding regions, which is reflected through the first synthesis of mRNA, and we're only interested in the coding regions. So I'll represent the coding regions by these shaded rectangle boxes, and they are officially known as exons. The non-coding regions are called introns. An example of those are the promoters and terminator regions mentioned earlier. To remove the introns, spliceosomes are used, and what they do is bring the exons closer together. By doing so, it kind of bunches up the introns to like a loop-like shape, like so. And then once that occurs, the spliceosome cuts the intron at the 5' end. And once that happens, this kind of like stubby end joins together to the other strand, forming a laureate loop. The laureate loop then kind of just leaves the exons, and then they are joined together, forming a mature mRNA. However, it's important to note that exons themselves can also be spliced by the spliceosomes, so then alternating exons will be joined together. This then codes for a different polypeptide, and a different number of proteins can be produced. This is another form of gene expression. We'll go on to explain another way gene expression can be regulated. Transcription is very important because if you're able to regulate transcription, you also regulate gene expression. What I have drawn here is a nucleosome, which is just a histone protein wrapped with DNA. And it's very important to note the charges. Histone is positive and DNA is negative. So to silence the gene, what a histone does is supercoils the DNA, making it very densely packed. And because it's so densely packed, the RNA polymerase is unable to enter the DNA for transcription. So how this is regulated is by adding a methyl group to the histone, or by methylation, because this maintains the positive charge of the histone. So to express the gene, the histone uncoils the DNA, making it a lot loosely packed. This allows the RNA polymerase to enter the DNA for transcription, and this is done by adding an acetyl group or through acetylation, because this neutralizes the positive charge of the histone. They do have their own scientific name. So once here, when it's very supercoiled, it's called heterochromatin. When it's loosely packed, it's called a euchromatin. So to consolidate what you're learning, I included some 3 practice questions so you can understand what the IB may be asking regarding to these topics. So please pause the video here and try them out, and then I'll show the mark scheme at the very end. Okay, so this is the mark scheme. It does says nucleoside triphosphates, which is just another way to say the free nucleotides, because when they're free, they have 3 phosphates, but when they bind together to form the mRNA strand, they loses 2 of those phosphates. They say histones are a group of 8 just because the histone protein is actually like a group of 8 proteins that are just kind of stacked on top of each other. And here is the last one. Yeah, it's important to note which one is the antisense strand and where is the end of the 3' and the 5', so then you can work out which is the 5' of the mRNA strand, and then you can work out where's the 3', because the free nucleotide will go to the 3' of the mRNA strand. Okay, thank you.